Hello. As you're rolling in, we'll give it a moment for people to come in through the electronic door, and then we'll get started. Feel free to use the chat function to introduce yourself and where you're from. We'll be using that throughout to, talk to each other. There's a lot of knowledge in this room. And then use the Q&A, and we'll try and use that to answer questions either during or at the end. But in the meantime, feel free to chat in the chat function and to introduce yourself. People are still actively rolling in, so we'll give it another moment. We aren't going to do raised hand tonight because it's just too many people to allow people to speak, and we've got some good information to cover. All right. Well, give it one more moment. Let the numbers stabilize a little bit, but welcome, and we're glad to have you here this evening. All right. Just some real quick housekeeping one more time. We're not going to use the raised hand function to let people speak tonight, but please, introduce yourself in the chat, your name, where you're coming from, talk to each other in there. There's a lot of people with a lot of knowledge in here. The Q&A we'll try and answer during the talk, and if there's time at the end, we'll go ahead and do a few minutes of questions there. Thanks for joining us tonight. We're going to talk about the important part of propagating California native plants. I'm Anne-Marie Benz. I'm the Horticulture Programs Manager with CNPS, and if you come to these regularly, I'm usually here. Tonight we're joined with Kevin Allison from the Catalina Island Conservancy, formerly with Tree of Life, as I believe Propagation Manager, head of growing there. And I'm going to go ahead and stop sharing and turn it over to Kevin. Thanks, everyone.
All right. Thank you, everyone. Okay, I'm going to go ahead and share my screen here. Okay. All right. Thank you, everyone. And thank you, Anne-Marie. I appreciate the intro and the opportunity to speak tonight on a topic that I think is near and dear to a lot of people's heart. And I think one of the most fun parts about native plants and getting into horticulture is to grow native plants and propagate new individuals. So as Anne-Marie mentioned, that's what tonight's talk is going to be about. So this is kind of a introductory talk, so I won't go too in depth, although I will touch on some more advanced methods. And if there's time left over, we can get into some more of the questions to answer those types of things. But this also will touch on a lot of the work that I'm currently doing here on Catalina Island. So how I'm currently I am the rare plant ecologist for the island. And what that means is that my job is to ensure that the rare plants of the island here on Catalina do not go extinct. And in order to do that, I use a lot of plant propagation and methods to be able to overcome a lot of these challenges that we have here on the island to make new plants and have individuals available for conservation, as well as conservation, preservation, those types of things to ensure these species don't go extinct. So as Anne-Marie mentioned, my background, I did spend the last about decade or so, about 7 years working as the production manager at Tree of Life Nursery, where I was focused specifically on California native plants. And so it was an incredible experience, and it really highlighted, being able to understand these principles of propagation, understand these principles of plant science, and really the ecology of plants in general on what they do naturally to be able to grow them. And not only to grow them, but then as a production nursery, and I hope that we continue this trend where we supply a large market of native plants for the industry. But understanding these principles in order to be able to produce a large number of plants to supply the demand for native plants and help support our native ecosystems, even our urban spaces. So that's where I was previously, but then I shifted gears to a big passion project for me, which is to conserve the plants on Catalina Island. So I've been here about a little over a year now, and we have a lot of really rare plants. And these are plants that do really great. Also, these island plants do tremendous in coastal gardens throughout California and throughout the floristic province. So, for instance, this Dendromecon harfortii, or the island bush poppy we have here on Catalina. So, doing some trials on some experimentation too to increase and enhance some of these methods of growing this through experience at Tree of Life, but also tailoring to make sure that the wild populations here on Catalina do not go extinct. Because we are seeing that trend. We are actually seeing a lot of Dendromecon out in the wild go, and I think on the northern islands as well, but it's certainly the case on here in Catalina. What we're trying to do to make sure that this rare plant continues for everyone to enjoy. So we do have a nursery here on Catalina. This is the James Ackerman Native Plant Nursery. And this is where we do a lot of that work, where we go out, we collect a lot of seeds, we take cuttings. Right now we're doing a lot of acorns for Oaktober, we like to say. And we have some rare oaks here on the island. But a lot of this goes towards not just producing plants for restoration, but also to have a living collection as well, to ensure that we have an ex situ collection or ex situ method of conserving these plants by propagating them at the nursery for future restoration, or just to ensure that these, in my case, for the rare plants, to ensure that we have a stock of them available. And then we also supply to the community here on Catalina Islands as also a method of biosecurity as well. Okay, so this talk, just a little bit of an overview. I'm gonna go over some introductory type things. We're gonna go over defining some terms, so we're all on the same page of what we mean when we talk about biology and botany and these types of botanical terms. And then I'm gonna go into some of the basic and more traditional methods of propagation. So that's with seeds and cuttings. And then I'm going to also touch on some of the more advanced type of propagation methods that people may have not tried before, but I encourage you to do so. So things like growing plants, ferns from spores, or air layering. And another thing I'm really trying to get more commonly used in the horticultural world here in California is plant tissue culture. And so that's really overcoming some of the challenges that you would have with traditional methods of propagation. Okay, so first of all, let's start. I'm going to take you guys all back to your biology classes, whether in high school or whatever. So I hope it doesn't cause too much PTSD, but just starting off, what is biology? And biology is the study of life. And then more specifically, we have the term botany, which is the study of plant life. I constantly, people I hear will say, oh, you're a botanist or horticulturist, and kind of throw those terms around, not specific towards what is actually might be that method. So botany can be like saying a surgeon is a biologist, but it's a very specific thing that they do. And in that case, you would call them either a horticulturalist or a propagator. So in horticulture, the term, I think it's Latin, is, break it down, it's hortus and cultura, or culturis, which means to garden and to cultivate. So to actually cultivate, it's the art and practice of cultivating a garden. Now, we use that term a little bit loosely too to say a horticulturist, so it's more the techniques involved in growing or taking care or cultivating a garden, which does include sowing seeds and propagating. So it does include plant propagation. So what plant propagation is to actually produce new plants. And that happens through two methods, where there's sexual and asexual reproduction of plants to reproduce and create new individuals. And so that happens like this. So this sexual reproduction, you have a where there's actually an exchange of gametes, where there's actual, just like how we all got here with our parents who then produce new offspring. So that's the sexual means, and that usually goes towards the seed, or it does. And then there's the asexual method of producing plants, or taking cuttings, or a rhizome, or divisions. And these are all actual clones where you're creating new individual plants, but it's actually exactly genetically the same as the parent material, or that it came from. So it's a clone. This does happen naturally in some cases with plants. So a cactus is a great example of that, where a pad might fall off the cliffside of a coastal bluff, and then it can actually produce new roots and form a new individual plant. Now, but that plant is exactly identical to the parent material that it came from. And we can mimic these type of things with propagation, or we can also stimulate with hormones to make new individual plants. And I'll get into that in a little bit. But the first thing I want to talk about is the sexual means of reproduction. And so this is with seeds. And seeds are incredible things. And I'm a kid that grew up reading comic books, and I always thought of seeds similar to like the story of Superman. If you know the S on Superman's chest actually represents hope, and I think that's really apropos to what seeds are. They are seeds of the future. It's a hope for the continuing of that species, much like in the case of Superman. So if you don't know the story, bear with me. So Superman and his parents, Jor-El, put Superman, or Kal-El, the little baby Superman, into a vessel as the planet of Krypton was dying. And that's very similar. I like to think of it as like a seed. So the inside of a seed is a little baby plant, and it's the hope for the continuation of that future. So even though the environment might be changing or anything like that, or that individual plant is starting to go through its life cycle and die off, it then creates a seed to then move into future generations, a hope for the future. And so that's very much like a seed. It's a protected seed coat with a little baby inside and a packed lunch with all these carbohydrates, and then is transported to a new environment. And then through different methods, that seed will then pick up on environmental cues, produce a root radical that will then go into the soil, put out new solar panels, using all that food and energy from that seed to where then it can then be powered by the sun, which Superman also is, and then grow into a really strong, new, healthy individual plant and continue that life cycle. And you can actually test whether these seeds are viable. So this is kind of a general method, but it can be done at home, where you can actually float seeds, or it's called a seed viability test or a float test, where if there's a well-developed embryo within that cell or in that seed, that it will actually sink. So it's denser than water. And this only works with seeds that are real, that will produce a denser than water seed, which is a good amount. So it's a general tool you can use and to see if your seeds are actually viable and will grow a new plant. And if they're not, they'll float. So this here is from some bladder pods. And you can see there's quite a bit of seeds that are nice and viable, and they're a little bit darker. Ones that did not quite develop that embryo, and it's more just carbohydrates and other seed material, then it floats to the top. So if it floats to the top, then it is not viable typically. And then a lot of seeds and this is kind of counterintuitive, but seeds are generally, or a good amount of seeds have dormancy mechanisms within them. They'll lie dormant. And so you would think that any seed would want everything it can to be able to germinate, which is kind of the opposite, really. Really, these seeds actually have mechanisms to prevent germination until the right conditions are right. And so that's a great way for plants to be able to take cues or know it and not waste the energy of that seed or that opportunity until it's right, until success is much higher. So these seeds will lie dormant in what's called suspended animation. It's still alive. It's still a living little baby inside that seed, but it's very, very slowly using the energy within that packed lunch into that seed until it's ready to germinate. And so this is when you talk about, okay, what's the longevity or how long are seeds viable, really matters to just the biology of plants, how long they can maintain within a seed bank and be a resiliency in the ecosystem to be able to come back. And then when we're banking seeds ourselves, like trying to understand, okay, are these seeds still good or not? But when we want to go and germinate these seeds, we're going to have to mimic a lot of these environmental cues that are like little locks and keys and little puzzles for these seedlings. And as a propagator, and it sounds like there's quite a few in this presentation today that are interested in it, that's the funnest part. That is for me, is trying to figure out these puzzles that the plant may need, and then have the results of new individual propagation or new individual plants. So why do we have these dormancy? Well, like I said, it's to be able to distribute seeds further. It's to also minimize competition in one specialized spot, and also allow for them to be undisturbed within a seed bank until those opportunities are right. And so there's also methods with seeds to transport plants. So like with the poppy, they burst open, or some of the like Caesalpinia, or I forget what the Caesalpinia's common name is. I think it's bird of paradise thing. I don't remember. But milkweeds, they have specialized structures to carry seeds, those little cotton-like material on them. Coconuts, that's a prime example of how they float, and then also through an animal. So we say typically this is like wings, waves, or wind are all these kind of methods where the seed actually, or the plant, uses energy of the environment to be able to transport elsewhere, because they're stuck in the ground, right? So it's fascinating. So how do we overcome this dormancy? And it's generally between two different methods, and it can be a combination of these as well. But there's physical methods where you're either physically scarcifying the seed, or actually filing down the seed, or trying to break down that seed coat. And you can think as a seed travels down an environment, blown around rocks and things like that, that could slowly start to break up that seed. Chilling is another method, or also known as cold stratification. And this is a method that mimics the environmental cues, where it goes really cold in the winter and then bringing it out. And so that difference of temperature to when it warms up signals to the seed, hey, we just went through winter where it would be too cold for me to grow. Photosynthesis and all that would be different, until it's nice and start to get into warmer, better environment and more sunlight. And it's able to then trick it into thinking it's spring to then propagate and grow out of that seed coat. Another method is fire, which can be both physical and chemical, but typically it's physical as we talk about temperatures. But it's also might be, and it depends on the species, but also the char rate, or the charcoal and the smoke, all can chemically also signal to the plant that it's now it burned in a chaparral or a fire, and now there's nice open, and it tells the seed, hey, there's now opportunity to grow, and there's less competition. Nice open canopy where it was once dense. And these are signals to the seedling that it's now time to propagate. And so we can mimic these in a greenhouse or a nursery. And so I already touched on these a little bit, but chemical, you actually can mimic the stomach acid of an animal that would break down the seed coat in its gut, or a bird or something like that, and to be able to then release the seedling to be able to grow out of it, or the embryo to be able to grow into a new plant. Hormone stimulants, that also is another method. Gibberellic acid is typically used with stuff like that. And then also, I can't really see with these controls here what it's my notes, but charring. Yeah, so the actual charcoal, or the leftover remnants of that fire and leaf litter that burned, and also so you hear about liquid smoke. Those types of things are used as a kind of cue to germinate seedlings and those that are from those environments. Some plants don't need any treatment at all, and that part of the strategy is it's trying to put out a lot of seed to cover large areas. This one is so grasses are a great example of that. Many grasses are windblown, and they will throw out a lot of seed that are either eaten by birds and other animals, or they can just repropagate in large areas. Invasive annual grasses are great at that, unfortunately, in our environment. But here this is one that we just recently collected a lot of, the a Catalina grass, a rare grass that's here on the island. And this doesn't need any treatment. We just collect it, have the seed, and are able to sow it directly. Here's another one. This is the Scrophularia villosa, or a Santa Catalina Island figwort, or Catalina Island bee plant. This one too puts out a lot of seed that don't need any treatment. They can just be sown directly, and their viability might not be last that long, but their germination rate's really high with no treatment. And that's just the method of that this plant has evolved to be able to. It decided, I'm going to put out a lot of seed, and just a lot of those are going to not make it, but some will, and that's the method of it. But other plants have more adapted structures on their seed to try to encourage more germination and success. Oaks are a great example too of not really needing too much treatment. These are ones you got to collect. Huge carbohydrates. They don't store in seed banks, so you got to grow them that year. But this is a great example. So we have the island oak here, which is one of the large oak seedlings here, or seeds. But here's a great example. Doesn't really need any treatments. Some areas where oaks are get a lot of cold, so maybe up in higher elevation areas. We don't have it here on the island, but cold stratification, or putting them in kind of a moist soil in the refrigerator for a little bit, can help start to mimic those type of things and encourage and improve germination. So it all depends on the species, and that's, if you ever played with science and especially plants, the answer is usually it depends, and that's definitely the case with oaks. Some seeds, like the Catalina nightshade here, is a rare nightshade we have on an island, will produce a nice big fruit. So it's in the Solanaceae family, same as tomatoes, peppers, those types of things. So it has a nice fleshy fruit, and you're going to need to get those seeds out of there. So once these are ready, you can cut them up. And if you've ever made like a nice smoothie and had a lot of seeds at the bottom of it, threw strawberries or something in there, you know you have a lot of seeds at the bottom. And you can do a similar thing with that. You process those seeds in a blender. We put little protection around on the blade, so it's and that way it doesn't really damage a lot of the seed, but like a weed whip, or have like a little fabric in there, and helps with that. But once you have that kind of nice slurry, you can strain it and then let it dry out and really get and release the seeds like that. And then they're good to go. Or you can try to mimic the actual gut of a plant. So we have an endemic manzanita on the island that is a wonderful food source for the island fox we have here. And and so this is one way that we, in order to propagate them, we try to mimic the natural ecology of that seed by germinating them using this certain process. So here, if you didn't know, manzanita stands for is Spanish for little apple. And you can see we just harvest these pretty recently, and you can see they look like a bunch of little apples. But inside that, you have a few of these really, really hard seeds around a fruit. So all these little apples are actual fruits, and inside there's a really tough seed coat. I mean, they're hard as a rock. But you can see relative to that seed and that nice, really hard seed coat is an embryo inside. So you actually have a really low germination rate with a lot of manzanitas. And that's why that seed coat needs to be broken down, and it can take a lot of energy to do so. But knowing that island foxes like to eat the seeds, we can mimic that and actually dissolve that really hard seed coat using acid. So you can use a concentrated acid. Definitely be careful when doing this. It's a very corrosive, dangerous chemical, but it mimics like our stomach acid. So make sure if you're doing this, make sure you wear all the required PPE or personal protective equipment, gloves, everything like that. Even be careful of breathing, so it can gas out a lot of things too. So take all the precaution, but the science of it is you're actually dissolving that seed coat. You can see that here. It turns like a bit of a black material, and this can take hours until you actually wear down and that seed coat, and it's nice and able to germinate. So Rhamnus pirifolia, or an island red berry, we have plenty of them here on the island. Foxes like to eat a lot of the toyon during the winter, so it has those berries. But in the summer, they like to eat the red berries when they're nice and fruit. So it's kind of got this year-round pantry for the island foxes here on the island. And you see in the scat, all these red berry seeds with the seed coats nice and started to dissolve. And so we actually do it. We'll actually sow fox scat. One, just also curious on what, is the diet of a fox. And we work with our wildlife team, so to understand that. But also it helps us in our propagation. And it's just, we do take volunteers to help us at the nursery here on Catalinas. If you'd like to volunteer and help us, you're more than welcome to. But the fox was basically like a volunteer in that case and helped us to do all the pre-treatments for us. Lemonade berry is a great one to also that use this method. So it has a nice hard seed coat. Tastes like a sweet warhead candy, but you can tell it's a really hard seed as well. And so using sulfuric acid is a method to actually break down that seed coat as well, as if like any other type of animal in the ecosystem would eat that seed. It's a method to break down that seed coat for germination. Fire stimulation. So fires are quite prevalent in California, and we have fire-adapted communities like the chaparral. And so a lot of the plants in these environments require or use fire as a method for propagation. And so the island bush poppy, the Dendromecon horfordii, or the mainland counterpart, which is the Dendromecon rigida, or chaparral bush poppy, I forget what the other common name might be, is what we would consider a fire follower. So after a fire, you would see germination occur from the seed bank based on those signals of the fire that occurred for the plant. And it's a bunch of different cues. It could just be one of these. Either it's the temperature of the fire, the smoke, the chemical, maybe a combination of them, but all those involved towards being a part of the ecology and life history and natural history of the plant to be able to stimulate that seed germination. So we do this. We will actually take a tray, use metal, or if you have plastic trays, make sure you line it with aluminum foil or some sort of metal around so you don't melt that plastic. And you have a nice, well-draining soil. Sow your seeds in there. and you can lay a nice layer of pine needles. Work really well on this. They're nice and dry, adds a lot of air into it. But you can use other materials as well, dried oak leaves, whatever. But pine needles seem to work the best. So you have a nice kind of a liberal layer, a few inches or so, and you set it on fire. Now you want to kind of stoke this fire, so you kind of get a nice even distribution, and it can get hot enough and not too hot in some areas, but you get this nice kind of even layer where every seed there kind of gets an even distribution or even treatment, and kind of moving around and making sure that flame really covers the seed flat, and then they're good to go. And so now you start to get some germination. This is actually recently, I think last week, we started getting some germination of Dendromecum, or the bush poppy. And you can see all that burned material around it that's helping these plants grow. One of everyone's favorites is the Matilaha poppy, also another poppy plant, or in the Papaveracea family. But again, another fire follower in the chaparral community. And same thing, they love fire. So you can start to do the same type of treatment, and those fire followers will start to germinate. Okay, now into asexual reproduction. So this is cuttings. So by taking cuttings, this is Gloria. We called her the cutting queen at Tree of Life Nursery. If you ever bought a plant from there, and we've been around for over 40 years, she has basically the one person who has done most all the cuttings at that. So if you think, oh my gosh, billions of plants, and no joke, definitely a few million, have come from just her taking cuttings, and she's really in tune to the plant and of when is the right time to do so. And so when you take a cutting, you're actually taking a little bit of an individual part of the plant, and you are using a root hormone, which is a signaling chemical or signaling compound, to signal to the growing parts of the plant to produce roots. And by doing so, you then have a complete plant with roots, shoots, leaves, all intact, with a vascular system to then continue on as a new individual. So from that, it's really important to understand meristems. And what meristems are really the growing points of plants. This is where cells are actively dividing, actively reproducing to new cells and creating new tissue. So you have your apical bud, which is up here on the top, or your apical meristem. You also have axillary buds or axillary meristems, and that's what you see along at each other individual node along a plant. You also have internally or lateral meristems as well, and that's what you see inside the plant that actually as they grow outwards. So if you ever see tree rings of a tree, that's actually each growth point, or the meristems, the lateral internal meristems that grow out. And I'll get to why that's important later towards propagation. But we also, move this down, have root meristems. So if you're doing cuttings of rhizomes, those are other meristems as well that go from that. So all of these to say that if you take a hormone and place it onto these meristems in a way that, or a cut, especially underneath a node, when you take a cutting, the lateral meristems there, if you touch with a root hormone, it signals to that rapidly dividing cells that, oh, that's creating new tissue, we need to create root tissue. And that's how you get roots stimulated to grow from a cutting. I don't know why it's not, there we go. And you can kind of see them here. If you look really closely, you can see the cells all in there, really packed and starting to really divide. So those are areas that if you were to provide a new a signaling compound or a signaling hormone, you would produce new roots. Okay, so this has been a great method we have the Southern Island mallow, also known as Malva rosa, here on Catalina Island. This is and these only occur on two little islets off of the island. And recently we went out and collected seed But we also collected cuttings because, well, one, it's just a diversified strategy. So that way, if the seeds don't germinate, we still have the cuttings available as well. But we want to make sure that this rare plant, it's only these 2 small populations on the island, don't go extinct. So we take a lot of cuttings around, enough. We don't want to take too many to where we make an impact, a negative impact on the population, but a representation of that population, and bring those back for propagation on the island. Malva does really well from cuttings, so that's why we do this as well. And then when we get back to the nursery, we take those cuttings. These are relatively big cuttings. They do well that way. And you can stick that, place the hormone in them, and eventually you'll have new roots forming from them. It's really cool to see that the flowers will actually still produce while the cuttings are growing, which is fun. But it also does well from seeds as well. So in fact, we had the seeds germinate from one population, but not the other. I don't know why yet. I got to figure that out. But at least I have the cuttings going. And so I have the genetics of that and those plants still going to make sure that if anything were to happen, we still have our bases covered. With Malva, they like actually their seeds germinate really well with hot water treatment. So in order to do that, and if you think, if it's growing out on this rock in the middle of the ocean or out in an islet, it gets splashed with water and things like that. And that water may even fall into the water, hit on a bluff or something like that. And that is soaked out in the sun, and that warm water, it helps soften that seed coat, and then the seed can germinate. So it uses hot water just really well as a seed treatment. And you literally would just boil water, place the seeds in there, and let it cool for like overnight, or just let it naturally cool down with it, soaking it for about 12 hours, at least 12 hours, 12 to, or overnight for 24. You can trial it at home, see what works best for you. And maybe the development of that plant at that time. But this is another treatment that you can do called hot water treatment, and it works really well with the Malva roses. Another method of cloning is division. So this works really well with bunch grasses or grass-like plants like the juncus or the reeds. But we recently went and visited the stream orchid, or the Epipactis gigantea. And this is one that this is all one plant here, all connected by a rhizome or rhizome root structure. And so the seeds weren't really ready, but we were able to collect rhizomes from this population. We only have one that we know of on the island, so we want to make sure we preserve that or conserve that. And we dig up a rhizome, bring it back, and let them start to repropagate asexually. So there was no seed germination. And you can see new rhizomes are then growing from those points, and then start to put out a new shoot, and create more energy, and create more rhizomes to continue. And we'll keep a nice stock of these growing continually at the nursery to ensure that if anything were to happen. There was landslides through there. These big rains, we were a little concerned about it, make sure that didn't wash away or damage the population itself. And we have a nice stock of it available for if anything were to happen to that population. Another plant that's really fascinating is the ironwoods. So we have a lot of people in this chat. I just want to really talk real quickly about a misnomer that's common in the horticultural trade with native plants. Many people call the northern island, or the Santa Cruz island, or the fernleaf ironwood Catalina ironwood, and that's incorrect. So that is actually a different subspecies that occurs on a few different islands, but up on Santa Cruz is one there that has a real fern-like leaf. And we have a Linothamnus, or a Linothamnus floribundus, but we have a different subspecies here that's endemic to Catalina Island that only occurs on Catalina Island. And that has a straight leaf. And so if you see a fern leaf, it's not a Catalina ironwood. You can call it a fern leaf ironwood, or a Santa Cruz Island ironwood, or a Northern Island ironwood, but it's not a Catalina ironwood. But this is a really fascinating plant. It used to occur almost widely distributed throughout all of the Western United States. I think the furthest fossil record of it was Utah, I think. But it had a long and wide distribution. But as climates changed over time, it sought refuge and now only occurs on the Channel Islands. The Lanthanus floribundus. Now, so it's since radiated into two different subspecies, but we have it here, and then we have an endemic subspecies here only on Catalina. What's fascinating, though, is that we have 120 groves here on Catalina, and there's a lot of trees in each one of these groves, but genetic research has shown that there's actually each one of these plants in these groves are clones of each other. So we thought we had thousands of trees, turned out to be only 120 plants, individual genetic plants, which is rare. So that's now a concern for us. And as the species name implies, floribundus, it puts out an abundant amount of flowers, but it doesn't put out a whole lot of viable seed. And we're trying to figure this out. One thing is that it's probably these seeds are self-incompatible. So you have so if there is only one genetic clone throughout this whole grove, that it's not producing a lot of viable seed because it needs another individual. Now, what are these too far apart? The groves are too far apart. Is the pollinator extinct or just in low abundance? And if we start looking into pollinators, I really want to. So if there's anybody that's an entomologist studying plant animal interaction, I would love for you to contact me. I'm looking to bring more of that type of research here to Catalina. But all that to be said is that this tree has basically lost a significant way to reproduce, which is by seeds. So it's struggling. And so it has a method of asexual reproduction, which is through basal resprouts or suckers or keikis, we call them in Hawaii. But this is a way that this tree can reproduce and create new tissue, new healthy tissue to hopefully keep putting on flowers. And over time can radiate out further and further into large material and more individual trees. And this is a method that it's just hanging on and continuing to make sure that it can not die off. And eventually that starts to, trees there can die. And it looks like many different trees in a grove, but they're actually all one plant. So we've tried to propagate them from seed. We get very little to no germination. We do have a problem with non-native invasive ungulates here on the island, which eat all the seedlings if we are able to get one. So that's a big challenge. And that's the rare case that you actually do get one to germinate. And it happens, but unfortunately that's the case. So what we do is we try to mimic this method of propagation, and we're using this more advanced method called air layering. And this is a fascinating way to grow plants. You actually will be able to passively create a large cutting of a plant, living part of a plant, or we'll scrape off the bark of the tree in a little section, right? We're getting down to that lateral meristem, and then we'll apply a root hormone there. And so it's that meristem is being stimulated by that root hormone to produce new root tissue. And then we pack it with like a sphagnum moss or a wet, basically a moist sphagnum moss. Cocoa core is another way to do it. And then pack that around in a protective layer to where the plant actually can supply water through its roots to that area, nutrients as well. It's still attached to the plant, where it can then passively start to create a bunch of roots where you can come back later, cut it off, and you have a whole new branch that's actually a whole new complete plant and a whole new tree. It's pretty cool. You can basically circumvent like years or many months to a year of propagation just by using this method. So here's what we did. So this is recently with the ironwoods, we're going out. You can see here's some of the plant technicians here that have stripped away some of that bark. You have some nice healthy growth on that branch where they've applied that root hormone and some of that sphagnum moss around, protecting it, keeping a nice tight seal around it so it's nice and healthy. It doesn't dry out. So there you go, nice and tightly packed. And then you put a little bit of a aluminum foil there, so it helps with the temperature and keeps it nice and healthy, where we can passively come back many months later and hopefully have a new plant. This is a new method we're trialing here. At Tree of Life, we did this a lot with Mahonia or Berberis. The Golden Abundance, that was a one that's a cultivar or a really attractive one that does well in a garden. Oregon grape, I think, is another common name. And this method does really well, where it was kind of challenging by traditional cuttings, not really great from seed, especially the ones that we want that particular traits of the cultivar. We wanted to clone that, and we would use this method to do it. We would call these tamales. As you walk by, and be like, okay, does it work by tamales? And we go harvest these tamales, come check them out, pull them out, see a nice healthy rootstock all in there, chop them off of the mother stalk there, and pop these up, and works great. It's a great method. A lot of tropicals, especially ficus, does well with this too. So if you guys want to try that. And then fern propagation. There's a lack of ferns in the horticultural trade, native ferns. And so this is something we were trying to do at Tree of Life, where we were trying to supply that. So these are older plants. Going back to biology, they have this fascinating method called alternation to generation. And so just real quickly, the life cycle here, well, it goes to a gametophyte and then self-fertilizes from the gametophyte and then produces the whole sporophyte. Now, this happens with seeds of other plants that are contained within it, but it's in flowers. But on ferns, it's actually two individual parts of its life cycle, almost these two different plants. So here we went out and collected some giant chain fern and got some of got those spores out of it, isolated those spores using different filters. And then here you go. Just like here's just like a little piece of a propagative soil media, and you can see these little flakes of plant. Those are actually gametophytes. And so the sexual parts are actual, like I think they're actually called sperm on the plant, will then have the male and female parts match on this little gametophyte, and then the whole, what we know as the fern leaf structures, will then come from that. Fascinating. And we call that alternation of generation. This is one here I think we also did with Polypodium, or the California polypody. That's one of the ferns that you can do by division as well. They do have a rhizome structure, or you can do it by spores. So two strategies there. The Woodwardia doesn't really do good rhizomes. It's more of a fiber structure. So here we had to do spores. And then lastly, one of the plants here on the island is we have the rarest tree in all of North America here on Catalina. This is the Catalina Island Mountain Mahogany. And this plant has a lot of challenges with it. So it's very rare. So we have this real genetic bottleneck. It's done so by a real history of ranching on the island. So a lot of the seeds have been eaten. And just limited to one population here in a really rough area called Wild Boar Gully. And this actually is a part of a conservation puzzle called genetic swamping or hybrid swamping, where you have these 6 remaining individuals in existence surrounded by a common mahogany, a common island mahogany, that has a lot of this pollen pressure and genetics that are causing nothing to come up but hybrids. And that's a problem because over time, it can basically be bred out of existence. And so this plant has basically lost its ability to produce seed. And if anybody's tried to do cuttings of a mountain mahogany, you have very little to no success. And if you do get them to root, it's very challenging to get them to be up-potted to a larger size and keep that plant alive. So this one can be rough. We've tried air langer as well and haven't really had much success. So what do you do? And so this is where a method called in vitro propagation or micropropagation, also known as plant tissue culture, where we grow these, this talk is called plant babies. So we grow test tube babies in a lab where we provide everything the plant may need to overcome a lot of those the challenges that may have the seed or cutting die or the plant material to die off traditionally. So we provide, it's actually like a nutrient gel. In vitro means in glass. So we have it nice and sterilized, all the glass and jars and nutrient gel and materials, all the vitamins and macro and micronutrients, everything the plant may need. And we find the right combination that optimizes that growth to where hopefully these little micro cuttings or each individual node of that plant can then start to grow root, and we'll really baby them gently in a lab, and hopefully then develop a new complete structure with it, shoots and roots and like that. What's great about this method too is you can actually get to rapid reproduction. So where you can have or exponential replication, where we can take cuttings off of cuttings. We can also go to this tissue mass and regenerate new plants from that. It's fascinating. And you can keep multiplying new individual plants from that as well. And each time you replicate these plants, and in this case of the Cercocarpus, where we only have 6 plants in existence, we're trying to clone every individual. And that way we capture every genetics possible. And then we're going to recreate groves with each of those 6 around the island to make sure that they don't go extinct. So that's what we're currently working on. Tissue culture is a fascinating thing. And if you guys have more questions about that, I'd love to answer those. But yeah, so we're working on the media now, and we have some great new shoots going. And what's great about this method is it's also a way to, like seed banking, which the CNPS is doing a lot of great work on that. We're also trying to develop a living bank of plants to where if you can't do seed, or bank seeds, especially like oaks, we can start to bank living plants on a lab. So like in the case of the island, the Catalina Island ironwood, we can actually have every one of those 6 or 120 plants in test tubes on a shelf in a lab, and the entire genetics of the species is there preserved, like we're trying to do with Cercocarpus as well. And so these are repositories that we can then go towards production and grow them out in a nursery, or we can also maintain them in a lab, much like you would seed banking in like a cold storage or something like that. So that's also the intermediate step to cryopreservation. And we're trying to work with San Diego Zoo to do that, to make sure that we can start to save these plants and preserve them indefinitely. So yeah, great method. It puts a lot of minimal stress on the plant while also producing a lot of material conservation. So all these to say that these are all strategies, seeds, cuttings that work great for conservation, but also towards the production of materials for the garden, which is, I think, also a great method of ex situ conservation to ensure that the plants of Catalina and California and all these native plants don't go extinct, and we help support our the natural ecosystems around in our environment. So I thank you. Here's my email if anybody wants to email me, and we'll turn it over for some questions.
All right, well, thank you, Kevin. We've got just a couple minutes, and this is recorded for everyone. It will be sent out in a couple of days, along with with some links for more information. And there are, as you can imagine, there are a lot of questions.
Mm-hmm.
Some of them are broad, some of them are very specific. But I'm going to start with a couple that are kind of foundational. Can clones produce viable seed And do you have recommendations for if propagating a certain percentage of seed for genetic diversity? Should more nurseries and home propagators aim to grow more from seed for that? So
Yeah. So when talking about cuttings in general, or cloning in general, whether it's by tissue culture or other methods, it is not a way of doing genetic diversity. However, the method in which you collect, and so that's what we do when we do cuttings in ways for conservation, is we try to collect from a diverse amount of different individuals to have basically a representation of that population with cuttings. So there's careful ways you can go about using cuttings in a way with native plants in production that still has genetic diversity, instead of doing an entire crop off of one individual or a couple individual plants. So that's more of a careful strategy and with that to the propagator to and I'm glad that you asked that question to be able to think about those types of things. Glad you're doing that. In the case of the Cercocarpus, that's one where actually instead of doing seeds, I would only be capturing a small amount of the genetic diversity, and cloning every one of them is actually capturing all the available genetics by cloning. So it's all a matter of it depends, like I mentioned earlier. If you're doing like cultivars, which are, I think, great. There's a lot of controversy a lot with the native plant world as far as that. But there are a lot of things when it comes towards different plants that can be of benefit. And you would want to clone those to keep to keep those traits available. And so that's where propagation using cloning is a way to do that. I hope that answered the question.
I think that got to a lot of it. Kind of a couple of other big ideas. When you're doing cuttings, does it open up the individual to disease? Does it matter the time of year? Are there best practices around that?
I think someone asked if micropropagation could also produce viable seeds. Yes, but again, it all depends on the strategy and the plant itself on which do and don't. And yeah, so that's the answer to that question. To this one, as far as disease, well, actually, a lot of plants are cloned for their disease resistance. So this is a lot of ones like tissue cultures use a lot for producing disease-resistant root stocks of plants that then are grafted the other plant for that produce foods or other ornamentals or whatever it might be. Palo Verdes are actually used a lot in this way too. So there's different methods, so it all depends. Does it open up to other disease? Again, I guess that goes towards the genetic diversity and strategy towards a healthy robustness. And then, so that all depends. That all depends on it. You wouldn't really want to use a lot of cuttings for restoration if you're doing a big restoration project. But if certain plants don't propagate by traditional means of seeds, you can ethically and through efficient protocols be able to capture a lot of genetic diversity by just collecting through a lot of different materials. And hopefully it starts to then self-propagate by asexual or sexually their non-produced new seeds and genetic diversity.
We had somebody ask, didn't someone do the in vitro part with Dudleyas?
Oh, that was me.
I figured that was a soft pitch.
Yeah, so that's what I did for my master's project. So that was partly not necessarily a restoration or a conservation of genetics. What that was using the principles of economics and using plant tissue culture to reproduce and clone a lot of plants. So it's a way to produce a lot of plants by taking a little bit of tissue from a rare plant. So these plants are already rare, and we could just clone a whole bunch of them, flood the market, and then actually through supply and demand, produce a lot of supply and decrease that demand to where there's no demand to poach a plant out in the wild. So it's a weird, through native plant horticulture and propagators, we can prevent a lot of these challenges and threats to conservation of rare species through cultivation. So yes, all these methods.
Well, I know we're not going to get to nearly all the questions. We will be sending this out with a lot of materials and follow up, but we are going to be having a real push towards making propagation protocols more available and finding ways to get this information out. Because if you're interested, we need a lot more people, organizations growing native plants and making them readily available. We want the industry to have as many plants as it wants or needs. So we'll be following up, and we're at time. So thank you so much, Kevin.
Oh, my pleasure.
Okay.
And yeah, if anybody, they may reach out to you, or if they caught my email in there, I'm happy to answer any question.
Great. And we had at least one note that they really like your sweatshirt. So
Oh. Yes. Excellent.
all right. Well, thank you, everyone. Thank you so much. Thank you, Kevin. And
All right. Thank
for
you
your
so
work
much.
as well. Bye.
Absolutely. Thank you. Bye-bye.